Okay, thanks for the introduction. Sorry, I'm unable to use the cameras. These Zoom problems I have. Um, but yeah, today in this talk, I want to share with you some of our latest results on control strategy that we have been designing and computationally validating so to engineer cell-fate. So we got inspired also as other worked on the Waddington energy landscape, which gives us like kind of like a course level intuition into how can we do cell-fate engineering. We can interpret that the cell-fate determination process that some cells could have follows some trajectory, right? That's governed by some endogenous gene regulatory networks, and it finally converges on some particular fate or phenotype. And so what we call classical cell-fate engineering basically tries to mimic this trajectory by using timely administered molecular regulators. So to try and replicate what happens right during development in vivo. And although these protocols methods work, they're not quite efficient. And that's where these biomolecular feedback controllers are come quite handy because it gives us this opportunity, right, to dynamically adjust directly the genes within those networks. And a very particular feature that's harnessed to design these kind of controllers is the fact that these gene regulatory networks exhibit multi-stability. So we can abstract these fates as the equilibriums and the stable equilibriums here in what's called the phase plane. And so if we want to make our system to end up in a desired fate, then we need to design more or less a controller that enables us to steer these trajectories so that they can converge to that desired fate. If we don't consider then one trajectory, but we consider a bunch of them, we can associate a probability distribution to each one of these fates, each one of these stable equilibriums. And so the overall goal of our project was to come up with a way to fine-tune these probability distributions. And before jumping on to the design, we come across some challenges. So the first one is that we were often working with partial knowledge on the dynamics of these gene regulatory networks. And so some controllers need some mechanistic insights for their design. And so that was the first challenge that we found. The other one was more related to current approaches related to cell-fate engineering, as we saw that they introduce some perturbations into the steady state of our process. That means that this equilibrium landscape gets a little bit disrupted and these ultimate fates, right, or stable equilibriums could eventually get lost. We took this as a design requirement also, and we ask ourselves if we can create a strategy to adjust the probabilities, but without affecting this equilibrium landscape. So essentially, we needed a controller whose effect was transient. And we encounter our solution when studying this, this network, which resembles an incoherent feedforward loop. So it's based on molecular sequestration. Here, U1 and U2 are able to bind and form an inactive complex. And there has been some extensive studies on this particular operation. And at the high sequestration regime, so very fast binding, this could be approximated to a subtraction. And if we just include explicitly an intermediate species, and we let the input to produce that species that then produces one of the sequestering molecules, and then that same input is able to produce the other sequestering species, then we're able to approximate a temporal derivative, a discrete temporal derivative, right? Because we will be subtracting the time-delayed version of our input and the input itself. These are the approximated dynamics that we got. Happy then to go in depth into the math, but I hope that you agree with me that if we just get rid of this proportional part, we will get kind of an ideal derivative, just coupled with a low-pass filter. So to do that, we defined an ideal derivative condition. It's called R. It's just a rate of parameters that should be equal to 1 so that we have this derivative operation. But why? Then why does a derivative will be helpful in the context of multi-stable systems? So it happens that if you remember those trajectories, as they approach their stable equilibrium, their values come closer and closer to a constant value. And so that time derivative will be closer and closer to 0. And since the output of our controller is proportional to that derivative, that means that it will eventually fade away. It will generate a transient perturbation, it will get to a maximum value, and then it will just eventually fade away. And so if we just imagine some dynamics of a gene that we want to control within that regulatory network, when we add this component, which is the effect of our controller on our network, we will see that the the additive means that theoretically we can then tune the probability distributions, and the fact that U1 converges to zero as also this derivative converges to zero enables us to do not affect theoretically, right, the equilibrium landscape. These are just some simulations to show you how the ideal derivative behavior looks like. So we kind of try to prove our hypothesis, this last hypothesis, by working with the toggle switch. As probably you are very familiar with them, but just in case, a couple of genes that express a couple of repressors that inhibit each other's transcription. When we test this, we analyze its steady-state behavior. We notice that it's actually a bistable system to stable equilibrium here. We ran some stochastic simulation from a sort of an equipotent initial condition, meaning that we will at the end get a probability distribution that's symmetrical and bimodal, of course. When we then use our controller in the manner that I'm showing here in the picture, you will see that we indeed were able to generate this kind of biased probability distribution, favoring the expression of one gene with respect to the other. And the phase plane is, or the equilibrium landscape, it's not, it's just barely perturbed, right? Initially, we maintain the number of equilibriums. We can further tune those probability distribution by means of the gain of our controller. So, of course, increasing the gain would lead to a bigger perturbation on the equilibrium landscape. So we wanted to address this just a little bit more systematically. And here we check on the location of the stable equilibrium corresponding to the Y1 gene, here to the Y2 gene. And you can see that, as we saw in the proximity dynamics, when you increase the sequestration rate, essentially capture again that property of not affecting the equilibrium while still getting the kind of bias probability distribution. And just as a final slide, we do acknowledge that setting these specific rate constants to be equal so that the complete adaptive metric is equal to 1 can be quite difficult to do experimentally. So we were wondering if we can relax this metric and how much can we relax that metric and still keep those properties that we have. So for this particular case, in our simulations, plus 10% error margin was admissible. We still were able to keep these biased probability distributions and also the equilibrium landscape within like 10% variation here. Also, that's what I wanted to present. The main takeaway of the talk is that we could harness this multi-stable feature that these gene regulatory networks related to cell fate have, so to design a derivative controller that gives us like an opportunity to talk about cell fate engineering, and that has this minimal footprint on the equilibrium landscape. And also, we find this quite interesting because it's kind of a new function related to the incoherent feedforward loop operation, which was right initially used for fault change detection and some newer work for those compensations. We think that this is a new contribution to the whole range of functions that the IFFL could have. And I would just like to thank Julia and Christian, as they were part of the team, and the whole laboratory, and some other colleagues whose insights on this project were really helpful to keep advancing it. That was what I wanted to share with you. Happy to take any questions.